Welcome to my session today. In the previous session, I have already given you to solve the sex determination in humans with the help of a Punnett square. Here is the answer. Sex determination in humans, it is XX and XY mechanism. XX male, XX a female and XY male mechanism of a sex determination seen in human beings. Here you can see the female with the 2 X chromosomes and 22 pairs of autosomes, and male with X and Y chromosome and again 22 pairs of autosomes. Female produces the gamete with the 1 set of autosome and 1 X chromosome. Both the gametes are of same type, so it is homogametic. In males, they present the X and Y chromosome. It produces 2 types of sperms, one with the X chromosome and 1 set of autosome, the other with the 1 set of autosome and Y chromosome. Now, you can see in the Punnett square the progenies, the different types of combinations. XX, that is female baby, and here also it is female. X and Y male. So, 2 female and 2 male progenies are produced. So, the sex of the child is determined by the type of sperm produced by the male parent. The same mechanism you can see here in Drosophila melanogaster also, where also the female is having 2 X chromosome along with autosome and male is having 1 X and 1 Y chromosome along with the autosomes. So, types of gametes produced by the male: A for autosome and Y chromosome, and A for again autosome plus X chromosome. So, 2 types of gametes are produced, hence it is male heterogamy. The gamete with the A plus Y chromosome, that is known as androsperms, and the gamete with the A plus X chromosome is known as gymnosperms. Female produces only ovum with the A plus X, autosome plus X chromosome only. That means it is homogametic. It is also seen in unisexual plants. For example, Melandrium album. Sperm with the A plus X chromosome, if it fuses with an ovum having A plus X, result in the production of female progeny. with a pair of autosome and one pair of 6 chromosome. Sperm with A plus Y fuses with ovum having A plus X. The progeny is AA plus XY, that is male. Now we are moving to the second mechanism of sex determination, XX and X0 mechanism. It is seen in some of the insects, for example, cockroach and grasshoppers and bugs. Here you can see female is again homogametic with the XX chromosomes, and male is heterogametic with only one X chromosome and no Y chromosome. So, it is written as X0. You can see here grasshopper, male with only one X chromosome, no Y chromosome here. So, we will return it as X0, female XX. Gamete, male produces two types of gametes again, heterogamy, A plus X chromosome and the other with autosome only. Female is producing only one type of gamete, autosome and X chromosome. So, female is homogametic. A plus X sperm when fuses with A plus X ovum, the progeny is female with the chromosomes AA and XX. The A plus 0 sperm, that means the sperm without any Y chromosome, when fuses with A plus X ovum, the progeny is a male individual. Now coming to the third mechanism of sex determination, that is ZZ and ZW mechanism. It is seen in butterflies, moths, fishes, birds and reptiles. Here the male is homogametic, homomorphic, represented by Z chromosomes, in order to distinguish from the XX male, XX female and XY male mechanism. Female is heterogametic, represented as ZW. Male produces A plus Z sperms only, that means only one type of sperm is produced, so that is homogametic. Female produces A plus Z and A plus W eggs. Two different types of gametes are produced, so it is heterogametic. A plus Z sperm fuses with A plus Z egg. Progeny is a male. AA plus ZZ combination. In the second case, A plus Z sperm when fuses with A plus W egg, progeny is female, represented as AA plus ZW. It is seen in fowls, chicken. Here we can see the combinations and gamete formation and all. ZZ is male, ZW is female. That means females are heterogametic and males are homogametic. ZZ and Z0 mechanism, just like XX X0 mechanism, it is at another mechanism of a sex determination seen in some of the butterflies. Here female is heterogametic, producing two different types of gametes, autosome plus Z chromosome and autosome only, that is represented as A plus Z gamete and A plus 0 gamete. Male is homogametic, produces only one type of a sperm, that is A plus Z, autosome plus Z chromosome. So, here male is homogametic and female is heterogametic. Now, regarding the fusion, A plus Z sperm, when it fuses with the ovum containing A plus Z, progeny is AA plus ZZ. It is a male homogametic individual, and the sperm with the A plus Z chromosomes fuses with ovum containing A plus 0, that means only autosome. The progeny is a female. So, females are heterogametic. Now, at another mechanism, interesting mechanism, that is Haplodiploidy mechanism of sex determination. It is seen in honeybees, wasps, sawflies and ants. Here unfertilized egg that develops into male individual. So, it is haploid having only one set of chromosome. You know the mechanism, if an unfertilized egg develops into an individual, then it is known as parthenogenesis. So, here the male individuals are developed from female individual by the development of unfertilized egg. Fertilized egg will develop only into a female individual. That means they are diploid. Females are diploid and male is haploid, having only one set of chromosomes. Sperms are produced by the mechanism mitosis only, because they are haploid. No chance of any reduction division. Whereas, since the female is diploid, ovum is produced by meiosis, reduction division. So, the gamete ovum will be always haploid. Here sex is determined by the number of a set of chromosomes. If it possesses only one set, then it will be male individual, and if it possesses two set of chromosomes, then it will be a diploid individual. So, here in the case of honeybees, in haplodiploidy mechanism, sex is determined by the number of set of chromosomes. Male honeybee has the chromosome number N is equal to 16, haploid. Female honeybee has 2N is equal to 32 chromosomes, that is diploid one. Male honeybee produces gametes by the mechanism of mitosis. Why it is so? It is because they are already haploid. Female honeybees produce gametes by meiosis or reduction division because they are diploid. Male and female gametes fuses and produces 2n is equal to 32 diploid individual, and we will get female honeybee. Here you can see the condition: female honeybee 2n equals 32 diploid undergoes meiosis producing haploid eggs. Eggs without fertilization, that is by parthenogenesis, it develops into male bees. Here again you can see the female bee produces eggs by meiosis, male bee produces the sperm by mitosis. When they are fused together, diploid female bees are produced with 2n equals 32 chromosomes. Here you can see the explanation again. Egg with n equals 16 chromosomes without fertilization develops into male honeybee. Here are some of the interesting facts. You can see male honeybees do not have any father because they are formed from unfertilized egg by parthenogenesis. Male honeybees can't produce any son. Moreover, male honeybee has only mother. Very interesting phenomenon. So, in sex determination mechanism, we have seen all these different methods: XX, XY mechanism, XX, X0 mechanism, ZZ and ZW mechanism, ZZ and Z0 mechanism, haplodiploidy mechanisms. Now we are moving to yet another aspect, that is pleiotropy. Pleio refers to many. A gene will produce many phenotypic effects. A gene will produce multiple expressions. For example, gene that control the flower color in sweet pea also control color of the seed coat and also the red spots in the axil of the leaves. The single gene here produces 3 different phenotypes: 1 flower color, 2 color of the seed coat, 3 red spots in the axil of leaves. So, this particular phenomenon, that we will call it as pleiotropy. It can also be seen in Drosophila. In Drosophila, the gene for wing size also controls eye color and fertility. Again, single gene that produces 3 phenotypes: wing size, 2 eye color, 3 fertility. Even we can see pleiotropy in human beings also. In the human disease sickle cell anemia, the gene alter the type of hemoglobin. It will also change the shape of RBCs. So, same gene here produces two effects, determines the type of hemoglobin and it will affect the shape of RBC. Again, in pea plants, synthesis of a starch is also controlled by a single gene. If it is homozygous and dominant, then it produces large sized starch grain. We are considering that particular gene as capital B here. So, capital B gene produces large starch grain. Capital B and small b, heterozygous condition, produces intermediate starch grain. Small b produces smaller sized starch grains. So, the size of the starch grain is controlled by single gene. Same gene that controls the size of the starch grain, which also controls the shape of the seed. Dominant and homozygous genes phenotype it will be round seed. That means capital B genes that produces the large sized starch grain, and as a result the shape of the seed becomes round here. In heterozygous condition also it produces round seed. Recessive and homozygous alleles genotype produced small sized starch grains. So, the size of the seed or shape of the seed that will be wrinkled. Here you can see yet another phenomenon also, incomplete dominance or intermediate dominance. Capital B, small b alleles together produces intermediate starch grain, and at the same time it will produce round seed. Now, we are moving to yet another important topic in this chapter, polygenic inheritance. It is also known as quantitative inheritance or multiple gene inheritance. Poly means many, multiple. Multiple gene inheritance, or it is also known as cumulative gene effect. Cumulative in the sense each gene, the effect of each gene is added. So, it is known as cumulative gene effect or additive gene effect. Here a character is controlled by many independent dominant genes. The inheritance of polygenic character is known as polygenic inheritance. That means inheritance of a character controlled by many genes is called as polygenic inheritance. There are so many examples: skin color in human beings, height in individuals, hair color in humans, weight in humans, intelligence, milk production in cattle, egg laying in chicken, etc., etc. So, in short, a character that is controlled by many genes is known as polygenic character or quantitative character rather than qualitative character. Why it is called a quantitative character? Because the number of dominant genes or quantity of the dominant genes will control the expression of this character, where each dominant gene shows additive effect. Each dominant gene has a specific dose in the production of the phenotype. So, when the number of dominant gene increases, the effect of the character will also increases. So, it is called a cumulative gene effect. In this chapter, we have to learn in detail about the human skin color, the typical example for polygenic inheritance. It is controlled by a pair of 3 separate dominant genes. We can represent the pairs as capital A small a, capital B small b and capital C small c. They are located on different chromosomes and they are inherited independently. If all the 3 genes are in dominant condition, capital A, capital B and capital C, the color of the skin will be dark color. The dark color of the skin is proportionate to the number of dominant genes present. If all the 3 pairs are dominant, then the individual skin color will be the darkest, we will call it as negro. A pure negro has 6 dominant genes for skin color and we can represent it as capital A, capital A, capital B and capital C. So, here 3 pairs of separate independent dominant genes are controlling the skin color, the maximum dark color of the skin. And at the same time, if these 3 independent pairs of genes are recessive, then it will be the pure white form. We can represent it as small a, small b and small c. A cross between pure black negro and pure white albino produces an F1 hybrid progeny with 3 dominant separate alleles and 3 recessive alleles, represented here as capital A, capital B and capital C small c. Since here in this case there is the presence of only 3 dominant genes, the effect of 3 dominant gene alone is represented as phenotype. It will be an intermediate one in skin color. We will call the phenotype as mulattoes. The skin color is intermediate between negro and white. That means between the darkest and between the lightest. In the F2 generation, since 3 independent pairs of genes are present, 8 different types of gametes will be produced, and in the F2 generation, hence there will be 64 different combinations. Here you can see the representation. A very light or albino represented by 3 separate pairs of recessive genes, and here very dark negro represented by 3 separate pairs of dominant genes. In the F1 generation, we will get only 3 dominant genes such as capital A, capital B and capital C, and 3 recessive genes small a, small b and small c. So, the effect of the phenotype, the effect of the or intensity of the skin color will be intermediate between both the parents, and the phenotype we will call it as mulattoes. So, in the out of the 64 different combinations in the F2 generation, when we are categorizing the progenies, we will see different phenotypes, different skin colors, such as 1 out of 64 will be pure black just like the parent, that is negro. Very dark brown will be 6 out of 64. Dark brown 15 out of 64. Mulattoes 20 out of 64. Light brown again 15 out of 64. Very light brown 6 out of 64. And pure white 1 progeny out of 64, just like that of the parent. So, here you can see the phenotypic ratio in a quantitative inheritance or polygenic inheritance as 1:6:15:20:15:6:1. It is easy for you to memorize. Write 20 at the center and 15 on either side, or you can memorize 15:20:15. Then on either side you write 6:1, or 1:6:15:20:15:6:1. So, this is the phenotypic ratio of a polygenic inheritance or quantitative inheritance. This phenomenon that was first studied by Sir Francis Galton. Here I will show you the gamete formation and you try to work out the F2 generation using the Punnett square. Here is the parental generation. The darkest phenotype represented by 3 separate pairs of dominant genes as AA, BB and CC, dominant alleles, dominant genes. Here it is recessive genes, the pure whitest form or albino, small a, small b, small c in pairs. So, 3 pairs of separate independent genes are controlling the phenotype in both the cases. This is the gamete produced here, capital A, capital B and capital C. Here the gamete is small a, small b and small c. When both the gametes fuse together in the F1 generation, when you are observing, you can see 3 dominant genes together and 3 recessive genes together. Capital A, small a, capital B, small b, capital C and small c. So, here the dose of 3 dominant genes will be there in the phenotype, and this phenotype we will say it as intermediate in skin color, that is mulatto. Now, here the gametes produced by this F1 generation will be capital A, capital B, capital C one gamete, capital A, capital B and small c the other gamete, then capital A, small b and capital C yet another gamete, capital A, small b, small c another gamete, small a, capital B and capital C the other gamete, small a, capital B, small c, small a, small b and capital C, and last it is small a, small b and small c. So, 8 different types of gametes you can see here. In the F2 fusion you will get 64 different combinations of genotypes and progenies, and we can arrange them in the ratio 1:6:15:20:15:6:1. Now, it is another interesting topic, that is pedigree analysis. It is very objectionable and difficult to do controlled breeding experiments in humans. So, we have to study about the inheritance of some traits in human beings by studying the family history. We have to visit the families, related families, members, and collect the data of a particular trait, how it is inherited, how it is moving to the progenies in different generations. So, it is done in many generations, not only in one generation. We have to collect a lot of data, then we will come to the conclusion, and it can be represented in the form of a family tree with so many branches using some of the symbols. Any Mendelian disorders that can be traced out in a family, how it is inherited or transmitted to the next generations, that can be studied with the help of pedigree analysis. For example, cystic fibrosis, sickle cell anemia, phenylketonuria, thalassemia, color blindness and hemophilia. All these characters, all these traits and their inheritance, that can be studied in a family by using this method of pedigree analysis. For representing the pedigree chart, there are so many symbols. So, we have to be familiarized with these symbols for making it. Male individuals can be represented by square, female individuals can be represented by circle, affected individuals can be represented by shades. This will be affected male showing the character under study, and this will be affected female. The affected individuals must be represented by shades. Then this particular line, it shows the marriage line or mating line. It shows mating between the parents. Now, these are the progenies, offsprings. Here it is the first generation after the mating, and this will be the second generation progenies. These two, this type of representation, it shows identical twins formed from the same zygote by cleavage. These two are non-identical twins. If in this case, if we are using a double line, like this, it will be the marriage between relatives, that means consanguineous marriage. Here you can see an example of a pedigree chart showing a dominant character, a character controlled by dominant genes, how it is inherited to the next generation or transmitted to the next generation. It can be shown, it is shown here. And if it is a dominant one, the character is a dominant one, then it can be seen in both the sexes. In male as well as in female it can be observed. Not even a single generation is skipped here, and it is vertical transmission. It can occur due to spontaneous mutation, 50% risk for affected child if parents are affected. So, here again about the representation of the male, female and all in the pedigree chart. Male represented by square, once again, female by circle, affected individual by shading, and here carriers are, here comes the point, carriers must be denoted by a dot at its center. For example, this is affected male and this one is, this is a carrier male and this is carrier female, not affected carrier. So, carriers are represented by putting a dot at its center. There is one more point. Suppose this is a shaded one, that means affected individual, and here it is written 2 at its center. It means 2 individuals are affected, and here it is written 5. It means 5 individuals are affected. Symbol with the number shows number of progenies. Line between the parents, again it is known as mating line, and double line between the parents shows the consanguineous marriage, marriage between the relatives, as I have already mentioned. Now comes to an important topic, that is sex linked inheritance. A typical example here we have to study is regarding haemophilia. There are so many other examples also. Color blindness is yet another example for sex linked inheritance. We have to learn hemophilia in this chapter. It is known as bleeder's disease. It is caused by a recessive mutant gene which is seen in the X chromosome, not in the Y chromosome. Normal gene for hemoglobin that is mutated to a recessive gene. So, in this particular patient, there lacks anti-hemophilic globulin which prevents bleeding, or it is needed for blood clotting. And in such cases, due to the lack of anti-hemophilic globulin in such individuals, there is profusive bleeding even if a minute injury takes place. It was first reported in Queen Victoria in the royal family. Carriers are not affected, but they will transmit this particular disease to their son. From son, the disease goes to granddaughter. That means from female carrier it goes to son, and from son to granddaughter it moves in a zigzag manner. So, it is also known as crisscross inheritance. Since there is the lack of anti-hemophilic globulin in hemophilic patients, even a slight or minute injury that causes heavy bleeding, profusive bleeding, and it can lead to death of the individual. Thus, hemophilia is an example of a sex linked inheritance. Inheritance of sex linked characters, that is known as sex linked inheritance. Sex linked in the sense the genes are linked to X chromosome. There are genes which are linked only in the Y chromosome. Such genes we will call it as Hollandric genes, and their inheritance is known as Hollandric inheritance. Hollandric inheritance will be transmitted from male to male, not from male to female. You know why it is so? Because the genes are present only in the Y chromosome, and Y chromosomes are transmitted from male to male only, not from male to female. So, the genes which are linked to the Y chromosome, that is known as Hollandric genes, and their inheritance is Hollandric inheritance. And genes that are linked to X chromosome, that is known as sex linked genes or X linked genes purely. We can see how the gene for hemophilia moves from parents to offsprings in different generation. This is a normal male with a normal hemoglobin. The allele is represented here as capital H, linked to the X chromosome only. Here in the Y chromosome, there won't be any allele for this particular trait. So, this male individual possesses only one allele for this particular character, hemophilia. This condition, that single allele condition seen in the male individuals, for a sex linked character, that is known as hemizygous condition. So, males are easily affected by this disease. Even the presence of a single recessive mutant allele leads to the production of the phenotype. That means they are affected individuals, they will be hemophilic. But see the condition of the female individual. Here you can see one capital H allele for normal hemoglobin and one small h allele for hemophilia, the mutated one, but this is a carrier, not a hemophilic one, because it carries one particular recessive mutant allele for hemophilia. So, it is known as a carrier. It is not an affected individual. See, this male individual, normal male individual produces two types of gamete here, one with normal allele on its X chromosome and the other without any allele for hemophilia in the Y chromosome. Here the carrier produces two different types of gametes, one X chromosome with a capital H allele and the other X chromosome with small h allele. Due to random fusion, there are different possibilities. One will be normal with both the X chromosome having normal allele capital H. This will be female individual and it is normal. And this is male individual X and Y chromosome, X with a capital H allele for normal phenotype. You can see both the X chromosomes are here, one with a capital H and one with a small h allele. This is a carrier, female. Here comes the affected individual, affected male. You know, see the condition here, only one X chromosome is present in the male and it is affected one with a recessive mutant allele. And there is no counter allele, dominant allele or any recessive allele in the Y chromosome. So, this particular condition, single allelic condition of the sex linked character, mainly X linked character present in the male individual, it is known as hemizygous condition. Now, you try to work out the same condition applicable for color blindness, where the allele capital C and small c present in the X chromosome of the female, as well as it can be seen in the X chromosome of male individual. Work out and bring it. Thank you for your attention.